Very pleased today to introduce Jenny Tung from Duke University. Jenny got her undergraduate and graduate degree from Duke, where she now resides, and then took a brief departure from that trajectory and did a postdoc at the University of Chicago under UCLA, and then returned to Duke as a faculty member. She, I guess, is renowned for being one of the pioneers of studying gene expression in natural populations. And gene expression is one of the unknowns. Most of us that study genetic differences between populations know nothing about what is often the immediate response to an environmental or social challenge, and that is gene expression. And she has begun with controlled experiments to kind of open that black box. She also is notable in the sense that she interviewed for a graduate position here at UCLA in 2003, turned us down, and she's one of the few people that have turned us down that has nonetheless achieved stardom. Without further ado, I'd like to introduce Jenny.
Thanks for that introduction.
Just to say, Bob, we understand why she turned you down.
I actually remember my interviews with both Bob and Dan really clearly, which was not why I turned it down. I'm not trying to say that. So it's a real pleasure to be here. My lab actually has some sort of informal ties to UCLA, so it's nice to actually be out here and meet the folks here and listen to the exciting work that's being done. I'm going to tell you today 3 different stories that loosely are connected by my lab's central interests, which are in social behavior and social structure on one hand, and various aspects of genetics and genomics on the other. To set the stage, this is a picture from one of the populations that we study quite intensively. So these are baboons in the Amboseli ecosystem of southern Kenya in East Africa. And I like this picture because, oh, that looks much better now. I like this picture because it does a pretty good job of showing you several of the different major dimensions of the environment that these individuals face on a day-to-day basis. So they're running across a grassland savanna, and these grasses are actually a really, really important nutritional resource for the baboons. In the dry season, about 50% of their foraging time is simply spent digging up these grasses to reach the underground storage organs underneath. You can see some acacia groves back here, and those are also a food resource for the animals. They produce fruiting pods that they use, and they also exploit the acacia tree gum. They're also sleeping sites for the animals, so they go up into acacia groves every night to stay away from predators on the ground. There's a Grant's gazelle back here, and we can think about that as symbolic of the large complement of other animals that reside in the Amboseli ecosystem, some of which are prey, some of which are predators, and many of which are competitors to the baboons. Now, of course, the thing that really pops out, I think, to us as humans is what's happening in the foreground. And this is probably the most relevant environment for this individual, too. And that's the environment that's being shaped for him by the male that's chasing him. So the thing that's probably the most salient to this male in the front is the physiological response induced by the social stressor of being aggressed upon. Okay? And we can imagine that his adrenaline levels are going up, his cortisol response is kicking in, and that if this kind of event occurs repeatedly over the lifetime, that it could potentially produce something more like a chronic stress response, which could have downstream effects on fitness-related traits like disease susceptibility and survival. There's some evidence for these kinds of effects. So here I'm showing you data from 2 papers that were published on this baboon population. On the left side are data from work that Robert Sapolsky did with my collaborators Jean Altman and Susan Albert some time ago, which shows that individuals, these are both males and females, who are lower ranking, that's on the right side of the graph, tend to have more dysregulated HPA axes. So they recover more slowly from the administration of a pharmacological cortisol than do individuals who are high ranking. On the right-hand side is from a paper that my colleague Beth Archie published a few years ago that shows that low-ranking males also take longer to recover from equivalent wounds and pathologies. So these sorts of patterns suggest that the stress of social subordination can be quite important in these animals' lives. This is a happier picture. So this is a bunch of individuals in our population sitting quietly in social affiliative bonds. And we think, based on recent work, largely driven by Joan Silk when she was here at UCLA, that these kinds of positive social relationships can also have pretty important fitness-related outcomes. So these data sets are, again, both from baboons, on the left side from Chacma baboons in southern Africa, where Joan first showed that females who are most socially connected, this is the top tertile up here, tended to live significantly longer overall than females who were less socially connected, and certainly than females who were most socially isolated in the social group that they were watching in Botswana. Recently, we published a paper that replicates these results with a bit of a larger data set. So these are females in Amboseli, and this differentiates those who have the closest social relationships with other females and with males from those females who are most socially isolated. So on average, the most socially integrated females, the ones who most successfully form these kinds of social relationships, live about 2 or 3 years longer than females on the bottom. And that's quite substantial. You know, that's equivalent to one offspring, given the average interbirth intervals of these females. Okay. In fact, this kind of work on non-human primates and on non-human animals in general is pretty far behind what's been done in humans. So observations that relate social adversity, and particularly social isolation, to health and survival have been reported in humans for many decades now. And what I'm showing you here is a figure from a recent meta-analysis that was published on 148 distinct studies that looked at mortality rates in healthy humans as a function of measures of social isolation. So all you need to know is that the further the bar is to the right, the bigger of a deal the effect on the left-hand side is. So in this meta-analysis, what they're estimating is that measures of social support and social integration lead to a 50% increase in all-cause mortality rates actually across both men and women and across all adult ages. So that's a huge effect. And in fact, we can sort of contextualize that by comparing it to the effect size estimated for a modest level of smoking and for a level of alcohol intake that's essentially alcoholism. So what they're saying here is that whether or not you're socially integrated into your community can have as big or potentially larger of an effect on the probability that you will die at any given age than smoking or a history of heart disease, which I'm not showing here, or alcoholism or obesity, okay, the big public service announcements. So I like to say that, you know, smoking is still bad for you. This is still clearly showing that smoking is bad for you. But if you're going to smoke, you should always do so in the company of friends. Okay, so this is a very interesting result, but it leaves a lot to be answered about what's actually driving this relationship between this non-physical interaction, right, social isolation, and its profoundly physical consequences. And there's been a lot of ink spilled on this question, and we still don't really know the answer. So to address this question in my lab, we take a page from Robert Sapolsky, who has long argued that if we want to understand social adversity, the consequences of social adversity in humans, we may be able to use non-human primate models. Okay, and these are the two non-human primate models that we focus on. We study rhesus macaques largely in captivity. This gives us the opportunity to do some experimental manipulation, and it also greatly facilitates regular sampling, particularly of slightly invasive samples like blood samples. And we couple that with the work that we do in this wild population that I've already alluded to of baboons in East Africa, which are able to tell us much more about whether this actually matters for individuals under natural conditions. So I'm going to tell you again 3 stories about these two systems today, focusing first on some work that I started in my postdoc on the relationship between social status, dominance rank, and gene regulation in captive macaques, and then moving to the baboons to talk about some more recent work we've done on how ecological and social variation and behavioral choices can affect genome-wide DNA methylation. And finally, I'll end with a story that should actually be coming out in print next week on how social relationships between individuals structure the baboon gut microbiome. Okay, so here's the framework that we're working in. So far I've told you that social adversity can affect what in humans is called health, and when we study non-human primates is called fitness, right? Because we're talking about fertility and survival related outcomes. And the way I've been talking about it so far suggests that the relationship between these two things looks like this, that you change levels of social adversity and that causes potentially changes in health and fitness related outcomes. However, in observational studies, including in humans, it's actually hard to eliminate some other possibilities. One is that this is all being mediated by health habits. I'm socially isolated, I'm going to eat potato chips for dinner, and I'm not going to exercise, and I'm going to take up chain smoking. Okay, so maybe this is not a direct effect, but mediated through health habits. Another possibility that's hard to exclude also in our wild population is what economists call a health selection sort of idea. And this is a reverse causation hypothesis, right? So females who are feeling sick don't feel like socializing. And so what you get then is the same kind of correlation between social adversity and these kind of health-related outcomes. Okay. So health habits, we can mostly toss out the window by studying non-human primates because nobody is smoking and nobody is drinking alcohol and nobody has differential access to health care. To get rid of the possibility that we're actually seeing reverse causation kind of explanations, we turn to these captive individuals. And in particular, we work on these rhesus macaques at Yerkes National Primate Research Center outside Atlanta, which we can manipulate in order to construct new social groups. Okay, how does this work? Well, in female rhesus macaques, normally a naturally constituted social group would have social status that's organized by matrilineal descent. So females would insert into the dominance hierarchy below their mothers. So you get these structures where high-ranking animals all tend to be from the same matriline, and then further down the hierarchy, individuals are also grouped by matrilineal descent. So we want to break that up in order to look at the effects of social status per se. And it turns out that if you remove adult rhesus macaques, adult female rhesus macaques, from their breeding groups and put them into new social groups, that the order of introduction into these social groups largely predicts the resulting social status. So females who are introduced into those groups first tend to be high-ranking, and females who are introduced last tend to be the low-ranking animals in those groups, and we can decide on order of introduction ourselves. We can form these new groups. In this case, we're using 5 females per group, and we can also replicate that experiment, in this case, in 10 different social groups. Okay? So this is just a picture of the group formation process, actually not from the particular individuals I'll be talking about, but from some more recent work. So these are high-ranking individuals, high-status individuals. These are low-status individuals. They're all starting at the same rank in the way that we're measuring rank here. And over time, actually fairly quickly, rank hierarchies emerge, and they tend to remain stable over time. So in my postdoc, we just asked this really straightforward question, which is, do these differences in experimentally manipulated social status affect the regulation of genes within these individual cells? So pretty straightforward study design. After the animals were organized into groups, we collected whole blood from these animals. We purified the peripheral blood mononuclear fraction, so this is essentially the lymphocytes and monocytes in the blood. And then we measured whole genome gene expression on, in this case, Illumina microarrays. For those of you who work with these arrays, these are obviously meant for humans and not for monkeys, and so we had to do quite a bit of quality control. We ended up looking at the expression levels of about 6,097 genes in the blood that we thought we could measure accurately. So the first question is simply whether there is a signature of dominance rank on sort of global patterns of gene expression, and we were very surprised to see how large of an effect we were able to identify. So what I'm showing you here is a relationship between dominance rank. These are high-ranking individuals. These are low-ranking individuals. And the first principal component of gene expression after controlling for differences between social groups and batch effects. And what we see is that rank appears to have a fairly pervasive effect on expression in blood. It correlates with this first major axis of overall variation. We can ask the same question on a gene-by-gene level to identify specific loci associated with rank, and that's what I'm showing you here. These are heat maps that depict gene expression levels for about 1,000 genes of that 6,000 gene set, where we were able to identify a relationship with dominance rank at a false discovery rate of 10%. So every row here is a female, and every column here reflects the gene expression levels in those females. What are these genes? It turns out that the biggest enrichment we see is for genes upregulated in low status individuals, which tend to be strongly enriched for immune system processes and especially inflammation, which is unsurprising to anyone who's familiar with this literature. Sort of constitutively high inflammation is characteristic of chronic social stress. We also see some sort of basic housekeeping and metabolic processes involved in genes that are upregulated in high-ranking individuals. So we did this experiment because we expected to see some kind of results, rank being such an important component of these individuals' day-to-day lives, but we were surprised about how large an effect we actually were able to obtain. And a natural question that arose then is, if this is so widespread, if I just looked at a gene expression pattern, could I predict the relative rank of the individual from whom that sample was obtained? So in other words, can I tell the difference based on gene expression data alone between high-ranking individuals, here we're considering ranks 1 and 2 high-ranking, middle-ranking individuals, or low-ranking individuals, ranks 4 and 5. And so to do this, we simply used a cross-validation type approach. We randomly kicked out 20% of our data set, so 10 individuals, and fit a model that related gene expression levels to the rank data we had, high, middle, or low, for the remaining 80% of the data set. We have this model now, ask how this model performs on the data that it never saw, right, on that 20% of the data that got kicked out. And we can do this 1,000 different times because you can kick out various combinations of that 20%. And so this is our result. It turns out we could actually predict rank pretty well. So across those 1,000 iterations, the median prediction accuracy for gene expression on relative rank was 80%. We got 80% of those females that the model didn't see originally correct. We also were able to ask at least a preliminary question about the plasticity of these kinds of effects over time. For a subset of our sample, for these 7 individuals, we had repeated samples, repeated RNA samples, when those individuals actually occupied different rank values. Okay, so in that case, we can ask, well, if a female changes her social situation, if she becomes high-ranking when she was before low-ranking or vice versa, do her gene expression patterns change in concert? Here we did very much a similar kind of prediction thing, except we used the 49 samples taken in at one time to train the model and then asked how it did on those 7 out-of-sample cases. And this is how we did. Of those 7 cases, we predict 6 correctly. If you guys have particularly sharp eyes, you'll notice that here I'm coloring them by ordinal ranks, whereas this is our rank classes that we're actually predicting. And if we look at only cases where rank classes were changed from high, middle, or low, we predict 4 out of 5 correctly. So again, an 80% prediction rate, very similar to the case that we observed before. So it seems like, at least on a global scale, we weren't able to look at individual genes. Changing your social situation also changes its reflection at the level of gene expression. So gene expression itself can be thought of as a phenotype. It's a molecular level phenotype, but like other phenotypes, it arises as a consequence of other kinds of underlying mechanisms and, of course, genetic and environmental contributions. So we spent a little bit of time in this paper trying to figure out why rank was influencing gene expression, and I'll just shortcut here and tell you that part of the explanation is perhaps a little bit boring. It has to do with tissue composition. So the relative proportion of different cell types in the peripheral blood mononuclear cells of low-ranking individuals differs somewhat from the composition in high-ranking cells, high-ranking females. And because cells differ in their gene expression profiles, we get this rank effect. Others appear to be explained by differences in glucocorticoid signaling. I mentioned earlier that a hallmark of chronic stress is often chronically high glucocorticoid levels and dysregulation of the HPA axis, and that also appears to explain some of these rank effects, which is a nice tie-in with much of the literature at the endocrine level. We were also curious whether some of the effects of dominance rank could be explained by an epigenetic mechanism, particularly DNA methylation. So why would we think so? Well, DNA methylation, as you guys are probably very familiar, refers to a covalent chemical modification in macaques, like humans, primarily on cytosine bases where they're followed by a guanine, so CpG sites. Okay? It is both stable over time, or can be fairly stable over time, and responsive to the environment, making it an attractive candidate for looking at mechanisms that underlie environmental effects on gene regulation. And in particular, we know that these things can be stable over time, that environmental responses can be stable over time, and can be important for downstream organism level traits. That's probably been most famously shown in this paper from Michael Meany's lab, published in 2004, where they were able to connect maternal care behavior with methylation levels at the glucocorticoid receptor gene in the hippocampus of rats, and that in turn predicted anxiety behavior in the rats, in those offspring when they became adults. So this is sort of the very crude hypothesis. Social environment affects the phenotypes that we care about. Maybe some of this pathway goes through gene expression levels, through gene regulation, and maybe the way that the social environment changes the expression of a gene is via epigenetics. So again, a very simple study design. Here we just simply could not spend the money to do all of our individuals, so we looked at 3 high-ranking individuals and 3 low-ranking individuals, again purified the same tissue type, PBMCs, and performed an extraction of the DNA and a conversion using sodium bisulfite, which means that Cs that are methylated still look like Cs, but Cs that are unmethylated get converted to uracil, which when you sequence the resulting data get read as thymines. Okay, so you can look at the same site, the same CpG site, and count the relative number of sites that are read as thymines versus cytosines to get an estimate of DNA methylation. Okay? And we just sequenced it because, you know, seems to be a lot of what we do these days. And we did the whole genome in this case at about 10 to 14x per individual. Okay, so this is basically what we were looking for. These are differentially methylated regions. These are rank differentially methylated regions. These are high-ranking individuals here and low-ranking individuals here. And each one of these little ticks is a CpG site in the genome. So at this particular location on chromosome 10, high-ranking individuals show higher levels of methylation than low-ranking individuals. And we find a bunch of these in the genome. Are these important? Do we care? Do they actually explain differences in gene expression? At a case-by-case level, we don't know, but we do know that these kinds of differences tend to be closer to gene transcription start sites that you would expect by chance, and also in particular closer to genes that are differentially expressed, that are rank associated on the level of gene expression, than genes that are not. In addition, if we use data on DNA methylation, we're able to perfectly cluster the low-ranking versus the high-ranking individuals. It's only 6 individuals, so that's not that impressive. But we can also predict differentially expressed genes from non-differentially expressed genes. So these are very sort of circumstantial pieces of evidence that suggest that that DNA methylation may be explaining some of the patterns that we observe. So does social adversity cause differences in health and fitness? We don't know still. What we think we know is that certain types of social adversity can probably play a causal role in affecting gene regulation. And this arrow on the right-hand side still needs some more work. We're trying to do that now. This is an ongoing project in my lab. We reformed 9 new social groups, and in this case, we decided to do what we kind of consider to be a more gold standard experiment, which is in the mid part of the study, we reorganized the composition of these social groups so that all individuals who were rank 1, for instance, found themselves in a social group together. Now, a hierarchy still forms from there. That means that some of those individuals who used to be high-ranking now are low-ranking. And some of the individuals who used to be low-ranking, who are all put together in some group, become high-ranking. So this maximizes all possible changes in rank in the first phase and the second phase of the study. We sample various things in the first phase of the study and the second phase of the study, which is just now completing. And we're asking about cell type specific differences now, instead of global PBMCs, actually cell type specific differences in genome-wide gene expression, genome-wide DNA methylation. We want to know whether social history, what you used to be before that switch, still carries a signal. And we're also asking about whether the rank that you occupy affects your response to immune stimulation. So we do this ex vivo using a variety of immune antigens, because that, we think, will give us much better insight into whether this even matters, right? Do these differences in baseline gene expression just go away? when you're actually challenged by your environment, or do they actually fundamentally affect your ability to respond to a pathogen? And finally, we're doing much, much more detailed analyses of different types of behavioral traits in these animals to ask whether things like being socially connected can offset being low social status. We are really in the depths of analysis, so I'm not going to tell you about those results here, but I will tease it a little bit by saying that overall, what we're tending to see is a much stronger role for cell types involved in the innate immune system than the adaptive immune system, and that most of the rank effects that we see probably maybe look like they are present at baseline and not in antigen-stimulated states. We'll see whether that holds. Invite me back in like 5 years and then I'll tell you. Okay, so this is all fine and good, but this is obviously a highly artificial situation that we're working with in captivity. And so my lab is really interested in asking the degree to which any of these kinds of relationships between environmental variation on one hand and aspects of gene regulation on the other carry through to natural populations where we can actually, think about a meaningful measure of say, fitness, which I'm not going to get to today. The population that I work on is this population in Ambicelli, where you've already seen some pictures from them. This is a longitudinal study that's been ongoing on a near daily basis since 1971. It was founded then by Jean Altman and Stuart Altman, who are at Princeton, and it's currently co-directed by Jean and Susan Alberts at Duke, Beth Archie at the University of Notre Dame, and myself. So this is a really lovely study population that I'm very fortunate to work on, because the depth of information on behavior and life history and demography is such that we can do pretty in-depth studies of the relationship between socio-ecological characteristics and, in this case, what I'll talk about today, DNA methylation. Okay. Most of this work is being done by someone that I think some of you guys know. This is Amanda Lea, my graduate student. If you guys have more like her, please keep on sending them to us, because she's fantastic. And she's really taken us in some new directions. Okay, how can we do this? Well, to measure DNA methylation, you've got to measure DNA methylation that's specific to a particular tissue type. And we're doing most of our work in blood. We can sample blood because although most of the work on Ambicelli is non-invasive, every once in a while we go out there and sneak around and dart these individuals. And that's actually what this picture is. This is Kinyo Waratere, who's a very talented field assistant who's been working with the project since the mid-90s. And these are a bunch of unsuspecting baboons who are habituated to our presence. You may not be able to see it, but there's about a meter-long black metal tube that he's holding like this, upright, in his right hand. And he's waiting for an opportunity when no one is looking. This is critical to avoid disrupting habituation. And he raises the blowgun, blows with considerable force, and launches an anesthetic-laden dart at a particular individual. In this case, we were darting a female named Luna. And then they go to sleep pretty rapidly. And when the animals move on, we pick up the animals and drive away and do kind of alien abduction kinds of things. I really like this picture in particular. Jean took this one because that's actually the dart. Can you see this? Like there's a little fluorescent mark there. That's the dart on its way to the animal. So it's a really, really good shot. Okay, so what do? Then we go back in the lab and we extract DNA and we measure genome-wide DNA methylation using an approach called reduced representation bisulfite sequencing, which again utilizes a high-throughput sequencing data generation platform as its endpoint. So this is pretty simple, and I'm happy to talk about this to anyone who's interested. It's a lot like RADseq, if you're familiar with RADseq, except here what we do is cleave genomic DNA with an enzyme that specifically recognizes a CCGG motif, right? So that means every end of every DNA fragment we get starts with an informative site, and we perform we make these into an Illumina sequencing library, perform bisulfite sequencing on the resulting samples, and then sequence them. This was all done at UCLA. In fact, we are very indebted to the UCLA Neuroscience Genomics facility because they were awesome about getting this protocol to work for us. We really like it because relative to just doing shotgun sequencing, relative to just doing the whole genome, we really enrich our resulting data set for parts of the genome that we think might be important. So this is the proportion of CPG islands, so dense clusters of CPGs that are important in gene regulation, and promoter regions that you would get if you just randomly sheared the genome, and this is what we get with RRBS. So this is a really good way to narrow down on the bits that you think you might be interested in, that we thought we might be interested in. These are simply quality control data. So most of the genome is hypermethylated. This is as expected. Most of the genome should be hypermethylated because most of the genome is essentially not active. And this is a relationship between our DNA methylation data and gene expression data. So this is from separate RNA-seq data that was just published about a month or so ago, where individuals genes that are most lowly expressed tend to be more highly methylated around the transcription start site, and genes that are more highly expressed tend to be more lowly methylated around the transcription start site. Okay, so our first foray into looking at differential methylation, which is what I'm going to tell you about today, focused on differences in resource abundance. So most of the individuals we study in Ambicelli are foraging in a completely natural environment, a pretty marginal natural environment, where the earth sometimes looks like this, and so there's not a lot of good stuff to eat. That's why they're digging those grass corms. And subsist on a pretty low-calorie diet. In fact, I learned recently that when Gene Altman wanted to study the consequences of this kind of diet in captive animals, it was considered sub-starvation level for animals held in captivity. In contrast, for about a decade or so, the project also studied baboons who were supplementing their foraging efforts with essentially scraps from a nearby tourist lodge. These were very high-fat, high-calorie diets, and they made for really fat baboons. So in fact, they spent half the time devoted to finding food. They spent 5 times as much time resting. They were much larger, 1.5 times body mass. And most strikingly, the body fat levels in females who live in their social groups their entire lives was 12-fold different between these individuals who are essentially eating what we eat if we go be tourists versus the wild feeding animals. They had higher insulin levels, higher cholesterol levels, worse teeth. So this difference in resource base was quite striking, which is why we started here. We figured if we were going to see anything, then we would see it here. Okay. So we can ask 3 questions. Do these differences in resource base predict genome-wide DNA methylation levels? Do these differences matter at all? Or is there any evidence that they're functionally important? And can they change? If males move between groups, as they do in the social structure of baboons, do their methylation levels, again, change along with them? Is their earlier life more important, or is their adult life more important in explaining variation in DNA methylation? Okay, this is actually a little bit of an advertisement. So we study these baboons in a natural population, and we have relatives in our sample set. And relatives mean that genetic correlations could also affect the variation in DNA methylation that we observe. So we didn't think we would have to do this, but it turned out we had to work out an entirely new computational method for dealing with the data that we produced. And so we did this in collaboration with Shang Zhou, who is in biostatistics at University of Michigan, and it's in a package that is called Macau. Basically, what we're doing here is taking into account of the fact that the number of methylated reads you see at every site is not only a function of the proportion of methylation, but also the total number of reads you see. So how reliable your estimate is, how much variance there is, depends on how much total coverage you observed at that site. Okay? And we can use a binomial mixed effects model to essentially relate the observed data to what we're really interested in, the effects we care about, in this case diet, and incorporate a random effect to take into account relatedness between individuals, or also population structure would work for that too. And we think this works better than anything that's out there right now. So this is a relationship between the number of sites identified as age associated in one of our baboon data sets, using 4 different commonly used approaches for modeling sequencing-based DNA methylation data. This is not very biologically interesting, so I'm just going to cut to the chase, which is that at any given false discovery rate threshold, we find more stuff than the other methods do. And we think that's because we're controlling correctly for genetic non-independence between the sample. Okay, so we used that method throughout everything I'm going to talk about. And if you're interested in using it, we'd be happy to share it with you. Okay, so what did we end up looking at? Well, we did a lot of filtering on our data set and ended up with just over 400,000 different CpG sites across the genome that we thought could be reliably measured and that didn't just suck away from our overall power. This is the distribution in different various compartments of the genome, including gene bodies and promoter regions and in CpG islands and shores and so on. Okay, so here's just the first pass question. Do resources, in fact, predict DNA methylation levels? So on the left-hand side, I'm showing you a QQ plot of the distribution that shows the distribution that you would expect under the null versus the distribution we actually identify in the real data. To cut to the chase here, essentially what I can tell you is that we found about 1,100 sites or so that were significantly diet associated at a 10% FDR. These are clustered together. They are found in the same places in the genome much more often than you'd expect by chance, which is what I'm showing you on the right-hand side. So this is showing the number of nearby significant sites when you center on any given one of these significant sites in the real data versus if you create an empirical null via permutation. And so we identify about 104 significantly differentially methylated regions as opposed to sites in our data set. Okay. So again, does that matter? Maybe these are just passive effects. Maybe they're just related to other sorts of cell types and they're not really functionally important in the blood. Maybe they reflect nothing about active regulation by this environment. So here's some data that suggests that maybe these are functionally important. So here we overlapped our data set with different compartments of the genome. So CpG island shores and CpG islands annotated by the Baboon Genome Project, enhancer elements, which are annotated by the Roadmap Epigenomics Consortium, promoter regions, this is just plus or minus 2 kb from 2,000 base pairs from the transcription start site, and other stuff. And what we find is that significantly differentially methylated sites are much more common in these regions of the genome that we would expect to be functionally important, CpG islands and shores and enhancer regions that are important in gene expression outputs. And they are under-enriched in regions that we have less reason to believe are functionally important, these unannotated regions. In addition, if we, again, cross-reference this data set with RNA-seq data, we find that differentially methylated sites are a little under twofold more likely to occur near genes that are actively being regulated, they're actively being transcribed in blood, and differentially methylated regions are even more likely. Now, I kind of skipped over the fact that perhaps surprisingly, sites in promoter regions are under-enriched for differentially methylated sites. That's probably maybe not what you'd expect, so that's kind of weird, and we think we can explain that. So again, using annotations from the Roadmap Epigenomics Consortium, this big attempt to characterize normal epigenomic variation in humans, we identified within promoter regions, plus or minus 2 kb from the transcription start site, 7 different chromatin states that were either over or under enriched for differentially methylated sites. And these are those 7, I know they're not labeled, but you can see down on the bottom 2 of the defining histone marks that characterize these promoter states. So the things that are over enriched, those are things that are marked with histone modifications that are suggestive of active promoters or of enhancer regions. So if you want to know what these actually are, so these are the ones that are over enriched. Their transcription start sites for actually actively regulated genes, not just promoters in general, but actively regulated genes, or enhancers near actively regulated genes, or enhancers near poised transcription start sites. Okay, what's under-enriched? These are repressed genes. This is why we're getting that under-enrichment of promoters overall. It's because we're including all of these genes that are not actually being regulated, and also gene bodies. Okay, because we're using that plus or minus 2 kb from the transcription start site, we pick up a lot of that stuff too. So in fact, promoters of actively regulated genes are pretty enriched for differentially methylated sites. Okay, again, that's pretty circumstantial, so it's nice to dig in a little bit deeper. We were struck by the fact that of the differentially methylated regions we observed, 51 of the diet-associated sites, the strongest signal was associated with one gene. Okay, and this gene is PFKP, phosphofructokinase, And overwhelmingly, if we look at all the CPG sites in that gene, each of these lollipops is a CPG site that we measured. They're all more methylated in wild-feeding individuals than they are in lodge-feeding individuals. Okay? What does PFKP do? Well, it actually encodes an important enzyme in glycolysis. It catalyzes this transition here, which is important because this is the rate-limiting step in glycolysis. This is the most tightly regulated step in glycolysis. This is actually an insulin-responsive gene, so that's kind of nice when we're thinking about a comparison based on resource abundance. And there's some evidence in the sort of GWAS literature that genetic variation at this locus is actually associated with obesity. So that's all kind of nice and circumstantial, and we still don't know whether these differences matter, right, to gene expression. But we can do an experiment, okay? We can ask whether these differences in DNA methylation at least impact gene expression levels, and we did that using two transfection assays, one focused on the upstream region and one on a region that includes some of the CPG sites upstream and some of the stuff in the first exon of the transcribed region. Okay, so this is how it works. This is a lot like a transfection assay that's used to determine whether differences at the genetic level produce differences in gene expression, except here what we're doing is cloning those little regions into a vector that contains the luciferase gene, so a glowing gene, except that this vector has been specifically engineered to have no CPG sites in it. It's a CPG-free vector that was a gift to us from the Relly lab. Okay, so then you can clone in your little bits of the promoter region of PFKP, and you can do nothing to it, right? And so in the process of replication, it's going to lose all methylation. It's a completely unmethylated case, and transfect it into a blood-based cell line. You can treat it with this restriction enzyme that recognizes a GC-GC motif, so that's going to partially methylate your construct at the part that you just inserted into it. Or you can treat it with this other enzyme, MSSS1, which recognizes all CPGs and completely methylates the region that you've inserted. Okay? And then you simply measure the resulting luciferase production and ask whether, as expected, completely methylated constructs express luciferase more lowly than completely unmethylated constructs. Does that make sense? Yeah, okay. So we just got these data like yesterday, and they look like this. They look exactly like what we had expected, which is the first time I've ever seen that happen in a transfection assay. So these are completely methylated constructs. These are completely unmethylated constructs. These are intermediately methylated constructs. And this is a measure of the gene expression of the reporter in the cells. We're going to do it again just to make sure, but so far it looks like this is validating pretty well. Okay. So maybe what's more interesting is to ask whether our data tell us anything about plasticity in the signature of diet. And again, here we take advantage of the fact that male baboons grow up in a group, but then leave when they reach reproductive maturation. And in our data set, we had 5 individuals who grew up in wild feeding groups, as marginal diet, on a marginal diet, and then moved. Yes? No? Okay. And then moved to lodge group, where I'm sure they were just, you know, what is it, pigs in hay or something like that. We also had 3 individuals who were born in this lodge/supplemented group and then moved to a wild feeding group. And we might expect that these changes have downstream effects on their physiology. In fact, Jean noted a while ago that differences between lodge and wild feeding animals were much less strong in males than in females. And that might be because of the persistent effects of nutritional conditions before dispersal from the group of birth, which would be a good explanation unless response to these new conditions is fairly complete and rapid. So that was nice to see. So how do we do this? Again, we came up with a predictive model for predicting whether an individual was lodge feeding or wild feeding based on the DNA methylation data. It works pretty well. Accuracy is about 79%. And then we tested 2 hypotheses. We said, for these individuals who switch, do they look like individuals that were in their natal groups, or do they look like individuals who are in the groups that they went into? Okay, so these are the 2 hypotheses: long-term stability of early life diet effects. So males who switch from wild feeding to lodge in green should look like wild feeding males, and vice versa. or a case in which there's substantial plasticity in response to shifts in diet. Is there a call somewhere? Where you actually look like the group that you went into. It's a cool call. Okay, so these are our two predictions, and this is what the results look like. And obviously, our imagination is highly constrained because they didn't look like either of our predictions. They looked like every individual who switched was predicted to be wild feeding. Okay, so if you had only looked at one direction of the switch from wild feeding to lodge, you'd say, oh, this is really stable. Early life effects are really stable. If you had had the other three individuals, you would have said, oh, these effects are really plastic. They change when you change your environment. Instead, what it looks like is probably interpretable in one of two ways. Either the experience of resource scarcity, of being wild feeding, just sticks with you no matter when it happens. Or, as Amanda has suggested, individuals who feed in a lodge feeding group over their lifetimes are just really, really weird. They're really different from everybody else, and we don't know the difference. But in some, we think that both diet during early development and diet afterwards probably has an effect on methylation, and that where individual males choose to go can have an impact on what their epigenome therefore looks like. We also think that our data really emphasize that it's important to be really contingent in thinking about plasticity. You know, when an environmental exposure could matter, but the direction of environmental change may also be really important to take into account. Okay. Last thing, on some recent work we've been doing on the gut microbiome, it turns out it's really easy for us to sample fecal samples, and you can do more with it than we ever thought you could. So why look at gut microbiome variation? Well, you guys have probably seen a lot of stuff in the popular press, and there's just a wave of studies coming out suggesting that what your gut microbiome looks like may be important in energy production, in metabolism, in defending you against pathogens or bacteria that have deleterious effects, and may influence your susceptibility to disease, especially autoimmune diseases like inflammatory bowel disease. Okay, so if this is so important, and that's not the side we've been working on, it'd be good to know what determines what kind of gut microbiome you're walking around with. What are the predictors of gut microbiome variation? We know a couple of them. What you eat is really important, and transmission from your mother is really important. Well, is there anything else? Well, one possibility is that social interactions may also shape the microbiome in a way similar to perhaps the way they shape the regulation of our own genomes. Okay, so this idea that social interactions might be important is not new. There's data from chimpanzees that suggests that there's a strong signature of social group on chimpanzee gut microbiome composition. And there's also data from humans that show that individuals who are unrelated yet live together in the same household, so you and your spouse, for instance, have more similar gut microbiomes than unrelated individuals who live in different households. You guys can probably immediately think of some alternative explanations for that pattern. In particular, well, maybe individuals who live in the same household eat the same steaks for dinner, right? And certainly, chimpanzees that are in different social groups actually tend to be foraging in different parts, different home ranges, mostly non-overlapping home ranges. Okay, so those are two different explanations for the patterns that have been observed, that social ties predict ecological or dietary similarity, contact, right, or exposure, or social ties lead to direct transmission. And we think that we can get at that in Ambicelli because we not only have these distinct and stable social groups, we also have stable and individually differentiated relationships within social groups. So we have two layers of social interaction that we can take a look at. So to do this is actually a pilot study that ended up being really interesting to us. We went out and sampled, we performed near complete sampling of all adults in two social groups that we study within 1 month. So we were trying to control for temporal or seasonal heterogeneity or heterogeneity in storage conditions and all that kind of stuff, right? And we imported them at the same time, extracted them at the same time, prepped them at the same time, multiplexed them together. So we were attempting to get them to go through as shared of a pipeline as possible, and generated about 30 million shotgun sequencing reads from each sample. Okay. We identified what was in the sample using a tool from the Huttenhauer lab called Metaflan. and also doing some de novo assembly. And we also estimated what kinds of, the relative proportions of different kinds of genes being produced by the bugs using another method called human. So again, this is really quality control. This is projecting our data, the baboon gut data, on data from the Human Microbiome Project, which documents microbiome variation in healthy human individuals. These are all from the States. And all you should really take from this is that human guts are more similar to baboon guts than human guts are to, for example, human nasal samples or human oral cavity samples. So guts look like guts and baboon guts look like a gut. So we first started by looking at between group differences, and what we find is that, remarkably, social group composition, whether you belonged to Micah's group or Viola's group, was the strongest predictor of what your gut microbiome looked like, and in fact explained approximately 18% of the overall variation in taxonomic composition. We see a very similar effect, although weaker, at the level of the enzymes being produced by these bacteria. So again, social group is the primary predictor and explains, in this case, about 11% of the overall differences that we observed. Okay, well, maybe that, again, is because of diet. These individuals inhabit a relatively homogeneous environment, and in fact, their home ranges, if you look at the year prior to sampling, do overlap. Nevertheless, it could be the case that relatively minor differences in what they're feeding on produce these differences at the level of the gut microbiome. And fortunately, we were also able to take a look at that by looking at our records on what they in fact were eating during the time we were sampling. Their diets were pretty similar overall, but we do see a difference in the proportion of fruit consumed between groups, with one of the groups, Micah's group, consuming a bit more fruit in their diet than Viola's group. So maybe that's enough, because we don't really know what's enough, to grade the differences that we observe. Because of that, we really relied a lot on our within-group analyses in which individuals were not, in fact, eating very different diets. They were moving around together in the same home range and exposed to the same kinds of resources. So these are networks that we built from grooming interactions for Micah's group and Viola's group in the year up to and including our sampling date, where heavier, darker lines reflect interactions between two individuals that are closer. They groom a lot, and then some individuals never groom together. They never touch each other. So these are measures of social affiliation that involve direct physical contact. So we can measure pairwise differences, dyadic differences in microbiome composition as well, using a measure from ecology, and then ask whether individuals who groom more together have more similar microbiomes. And in fact, they do. So these are the data from Micah's group, and these are the data from Viola's group. And in both cases, individuals who groom more have less dissimilar, i.e. more similar, gut microbiomes. And we see that in both groups for taxonomic composition, and in Micah's group also if we look at the level of enzyme composition. Okay. So again, you could say, well, maybe within these groups, individuals who groom together just happen to be exposed to similar microenvironments. They sit next to each other a lot. Or maybe they just tend to eat diets that are a little bit more similar than individuals who don't groom together because they're around each other a lot. So we were able to take that into account as well. So these are grooming networks, social grooming networks on the left, and microbiome networks on the right. And we could ask, okay, well, what if we create similar kinds of network structures for dietary similarity or for proximity, actually just being spatially close to each other as opposed to directly touching each other. So we used partial Mantel test to ask whether we still see a signature of direct social contact on the gut microbiome controlling for dietary similarity, and we do. And we asked the same question at the level of spatial proximity, and we still see an effect of direct social affiliation. This relationship also holds in controlling for kinship, if you're wondering. Okay, so that at least suggests that there might be a direct relationship between social affiliation and the gut microbiome. What are these species? We find 327, of the 327 most common species in the gut microbiome of baboons, 64 are differentially abundant between groups. Within networks, we use a metric from spatial statistics to ask about correlations between the social network structure and the abundance of particular microbes. And again, looking at the same 320 bacterial species, this is just an example, we identify, about 50 that appear to be structured by social networks. So that means that your close social partners are going to look similar to you in terms of abundance. These overlap actually quite a bit. So these are the enriched taxonomic enrichments for socially structured species, either between groups or within groups. And each of these red stars indicates enrichments that are consistent both at the between group and within group level. There's a significant overlap between the bugs we find at both levels of analysis, suggesting that some of the differences between groups may be based on the same sort of affiliative grooming mechanisms that we're picking up within groups. Okay, so social groups differ in gut microbiome composition. That's not new. What we think is new is our ability to use really fine-grained behavioral data to indicate that this process may be mediated by affiliative direct physical contacts between animals. So we think that this might be another path that's been relatively underexplored, to bring it back to my, my initial setup, by which social relationships might be able to influence health. And certainly, although it's hard to predict which direction, something that may be a potential cost or a potential benefit if they're sharing beneficial microbes of social group living. Okay, so just to wrap up, I've told you 3 things. 1, dominance rank influences gene regulation in captive rhesus macaques. 2, that ecological variation, we're also looking at social dimensions now, affect DNA methylation in what can be a relatively stable manner. And finally, that social interactions seem to influence the composition of the gut microbiome. And with that, I'm just going to finish with my acknowledgement slide. This has been work that involved a lot of people. The macaque work in particular started when I was a postdoc with Yoav and continues as a collaboration between Luis Barrero at Montreal, Zach Johnson and Mark Wilson at Emory, and myself. The microbiome work was a collaboration with Luis and with Ron Bleckman at Minnesota and Beth Archie at Notre Dame. All of the baboon work is joint between Jean and Susan, Beth and myself. And we owe you guys here at UCLA for being able to generate the DNA methylation data, and of course also for Amanda, without whom much of that work would not be done. And I'm happy to take any questions.
I want you to write a letter to our team to run out how much you like our work.
I'm really, really happy to do that. I actually, if we have time, I'd love to go say hi to Joe because he's been awesome.
But so I wanted to ask you about the lodge compared to the so how much behavioral difference is there? You know, just sort of looking at non-eating behavior.
Yep.
Just sort of being in that environment.
Yep. There are fairly substantial behavioral differences. Their time budgets really shift. So they're doing a lot more socializing and a lot less foraging. This was not the case at the time these samples were collected, but probably because of the resource abundance there, they, those social groups started growing really fast. And so the nature of the social groups were less stable as well. There were more fissions, we think, that happened in there since we stopped watching them. So there are quite a few things. We think they're driven by resource base, though. Yeah.
But I mean, how much do you think the behavior itself, sort of the overall behavioral driving?
Yeah, I think that's probably unlikely for the PFKP case, at least, because that's so clearly an insulin-sensitive regulated gene. It may be the case for some of the other sites that we're seeing. When we obviously have a sort of data set like we have here, where you have multiple things that are perfectly correlated with each other, that would be really hard to disentangle.
I mean, the genes that are responsible for human obesity are almost all, the ones that are insulin regulated, are almost all behaviorally driven.
Because human obesity is behaviorally driven. And so you could argue the same thing here, that these baboons, yes, it's behaviorally driven by their desire to eat tasty, fatty things. Yes, I mean, you could think about it that way, I think. So I suppose it depends on how far, back you want to go on the causal pathway. Yeah, good question. Yeah, Dan.
So ontogenetically, we're finding with the marmots that some things are transmitted to the next time step. So early childhood experiences may have a legacy as yearlings, but it may attenuate over time.
Yeah.
Whereas some things persist over time. Have you been looking at this developmentally at different time steps, or is it just one versus another?
So because our darting is opportunistic, we don't have it'd be ideal to have regular yearly sampling as these individuals grow up. We do have repeated samples for some individuals, but we haven't exploited them yet. What we are doing, so we have some funding to look at social status effects in early life and adulthood. There we're really using samples collected in adulthood where we know both their early life status and their adult status, and trying to identify which of those appears to be most important at what loci in the genome. My guess is that there's not going to be one answer to your question, right. And of course when you're looking at 400,000 things, yeah, exactly. But I think it's a really interesting one that we'd like to get at. So yeah, sure. Yeah.
So a question sort of spans the first two parts of your talk.
Okay.
I'm wondering if there isn't an analogy that can be made between health habits in humans and resource availability in non-human primates.
Mm.
So in other words, are low-ranking chimps just potato chips is all they get? So I'm wondering, is there some sort of, has anyone looked at a negative feedback loop between, yeah, you have the stress of being a low-ranking individual, but your resource availability is also very different in a natural habitat?
I'm sure somebody has looked at those questions in some species. So in the captive animals, they're all being fed the same thing.
Right. So
yeah, so that's controlled for. In the wild animals, when we think about why status has the effects that it does, certainly explanations that are mediated through resources are probably important. Now, that's not actually very easily measurable because if we look at the composition of their diets, it doesn't really differ within groups. What might differ, and it's something that I know Susan wants to look at, is how often their feeding is actually being interrupted. So, it's not, they're eating these grass corms a lot. It's not like grass corms are here and grass corms over there are better. Again, that's 50% of their foraging time. But if you're doing this, relatively energetically expensive extraction activity and you get interrupted a lot, that may have an impact on the overall resources that you're able to.
Things like that. I don't know if there's differentiation there for low-ranking individuals.
I don't know the answer to that because we can't see them then.
Right.
Yeah.
Yes.
Did you or would there be some use in actually using a high-throughput method to examine the food that these things are eating from the feces as well as the bacterial content?
Well, I don't know how good that technology has gotten. We know what they're eating because we write down what they eat. What we don't know is like a measure of what they're eating and exactly how much they're eating and what their relative caloric intake is from those different foods, because that's just a level of granularity that we don't do. Stuart Altman did a little bit of that, but it's like you can't do anything else if you're going to do that. So I guess my question back to you is whether you can actually get that from fecal samples.
I mean, my interest is because I'm interested in systems where you know a lot less about what's going on.
Mm-hmm.
If you could assay all these things without the effort of actually having all this field time,
yeah.
Then maybe you can learn about other systems in addition.
Mm-hmm. So we haven't done that, although we've been sequencing a lot of fecal extracted DNA recently. Most of what's in there is microbial. I don't know how much is plant material. We have not tried to look at those reads to identify food items. We could try it.
Jenny, I have one question.
Yeah.
So in the human microbiome literature, it's clear that there are various communities of microbes in the gut, some of which are pathologic or associated with health. And it's probably the same way in baboons. So what I'm wondering is if there are these different kinds of communities amongst a social group, then those that grew more, the popular individuals, the ones with a lot of centrality, are getting much more feedback of different microbiomes, all the individuals. So it seems like their health state might be different from those that don't grow as much and are stuck with their own microbiome.
Yeah, I think that's a really, really interesting question. We don't know the answer to it because in fact, that's related to one of the most immediate questions that came up for us when we saw those results, which was, well, is this good or is this bad? Mm-hmm. Do you want to be doing a lot of sharing or do you not want to be doing a lot of sharing? And ultimately, that probably depends on what you are actually sharing. And I didn't talk about it, but we did some work to try and identify what socially structured microbes were like. What's the lifestyle of the ones that we can see being apparently passed back and forth? And the most confident result we have, which is actually the only thing that went into the paper, is when we asked whether the microbes that tended to be shared were more likely to be anaerobic and non-spore forming, so really dependent on living in the gut. We thought about trying to ask that question from the level of pathogen, whether these bugs are pathogens, but that's so poorly annotated, and there are so many species that can be pathogens but aren't always pathogens. Like, most of us are walking around with E. coli probably, and we're probably fine, but it can be pathogenic, and that's a strain level and a host state level kind of phenomenon. So we really like to do something that I think is similar to what you're asking about, which is to say, is this good or is this bad? This one's good, that one's bad, this one's good, this one's good, this one's bad. But we can't.
I think it's more nuanced than that, though, because many of us are walking around with MRSA. We shake hands with lots of people.
Yep.
We don't necessarily transmit MRSA.
Yep.
Or we do transmit MRSA, but it doesn't break out.
Yep.
So it's really the ecological context of that gut microbiome. So this is just first steps, important first steps, in determining how those things are changed,
yeah.
understanding the sort of functional significance and how you change something from a healthy state to an unhealthy state, of which there are many pathways to do that, is virtually unknown.
Yeah, I think that's right. I mean, this was a pilot study. We're doing more now because we think it's interesting. But
one last
yeah.
Yeah. So again, on the microbiome. So maybe you said this and I missed it.
Yeah.
But I mean, what's the mechanism for you and I groom
right.
us having similar? Is it the stuff I pick off of you and eat? Is that the mechanism? What is it?
Okay, so it's a great question. Actually, this is
vice versa.
Yeah. Not to make it personal, right? So we think that probably does have a lot to do with it. So these are animals that are walking around on the ground. They're also defecating on the ground. They, in close affiliative contact, they are grooming each other. They're really looking through each other's fur. And for females who are in estrus, who produce these really lovely sex skin swellings, there's a lot of attention to the hindquarters in particular. So that seems like a likely method for fecal-oral transmission in this species. They're not coprophagic. So for other studies that have shown kind of social transmission in bumblebees and rodents, they're actually eating each other's feces. It's a really, really direct mechanism. That's not happening here. Although they do eat elephant feces for whatever reason. They do. Or they pick nothing, yes, but yes, they're doing all those things sometimes. Yeah. Mm-hmm. Yeah. We haven't yet. We've done some ectoparasite collection in years when the tick loads in particular got really high. They kind of go through flushes. And that was actually mostly to look at hematoparasites in the ectoparasites. We haven't actually looked at microbiomes in the parasites. I guess you'd look at the surface and what was inside them, and yeah. I'm sorry to end on that right before lunch, but.